In this lesson, we'll take a little bit closer look at mitosis. We've seen this picture before, a bunch of onion root tip cells that have been stained with a blue stain, and it reveals lots of cells are in interphase of the cell cycle. All the cells with nicely evenly stained nuclei are in the interphase stage of the cell cycle. Now, why is it so evenly stained? Well, inside the nucleus, the DNA is unwrapped, it's unwound, and so the staining doesn't give you the clumping that you would see over here when the DNA is all coiled up. So the even staining of the nucleus is because the DNA inside the nucleus is unwound. The clumping of the stain, where we see the colored bodies, the chromosomes, is because the DNA has been coiled up into that more compact structure. Well, we see most of the cells are in interphase because that's the longest stage of the cell cycle, and every time we see chromosomes, then we're in some stage of mitosis. Here we see a condensing stage, here's a lineup stage, this might be a lineup as well. Here we see a separation stage, here we see a division stage. Looks like that's close to a division stage as well. In fact, here it looks like you can see the cell wall here that is formed between these two clumps of chromosomes here. So in this picture then we have cells in interphase and cells in mitosis. So let's go through the process in more depth. The first stage of mitosis, the condensing stage, we named it, occurs as follows. The nuclear membrane around the chromosomes is going to begin to break apart as the chromosomes are condensing. Now this is important, of course, because if you're going to separate your duplicated chromosomes, they can't be trapped in the nuclear boundary, right? So the nuclear membrane actually breaks down, thus freeing up the duplicated chromosomes. Here we see an interphase cell, and so the nuclear membrane is confining all of the DNA inside the nucleus. So here we see the stain is clumping up because the chromosomes have condensed, they've coiled up, and they are now free of the nuclear membrane. Now, duplicated chromosomes, remember, are duplicated because the DNA molecule got duplicated, but they're still attached until they get separated during mitosis. In fact, here you see a picture. These are actual human chromosomes. And notice they all sort of look like X's. Now, that doesn't mean they're X chromosomes. It just means all of the chromosomes are in the duplicated state, but still attached, as in our picture. Here we see another bunch of human chromosomes. So this is it. These chromosomes are found in every human cell, and this is the information, in a sense, to make a human being. We know more specifically the information that chromosomes contain is the information to make proteins. But this is it. This is what it takes to make a human being. Now, something else very important also happens during the condensing stage, the first stage of mitosis. So this is an important stage. In addition to the nuclear membrane breaking down and the chromosomes, the duplicated chromosomes condensing, a set of proteins is going to arrange themselves. So microtubules are going to start to form from each side of the cell. Now, in the old days, they were called spindle fibers, and it's a bunch of different proteins, but microtubules are a part of the story. So we're going to have a kind of a scaffolding of proteins that will assist the cell as the cell separates its duplicated chromosomes. Here we see the microtubule organizing center, and we have microtubules of different lengths projecting upwards here. So this kind of thing would be on one side of the cell, and another one of these would be on the other side of the cell. Now, the job of these microtubules on either side of the cell will be to extend these microtubules and to find the duplicated chromosomes wherever they are. Remember, they were released from the nuclear membrane, so now they're sort of just kind of drifting around in the cell. Well, they have to be found in order for the chromosomes to be separated. So the microtubules are going to grow, and sometimes retract. They're going to be searching. Now, that's metaphorical, right? They're going to be searching for the chromosomes, and this is a trial and error process. And when they find a chromosome, they can sort of attach to that attachment site here. So here, this chromosome has been found by this microtubule, this chromosome has been found by this microtubule, etc. Now this stage here is still sort of condensing stage, where now we have a set of proteins. The microtubules are going to set up a kind of a scaffolding. They're going to search for the chromosomes and then help to line them up and that will be the second stage of mitosis. So the second stage of mitosis looks like this for a real cell, where we see the chromosomes lined up. We happen to have a cell next door that's in the condensing stage. And here's another one over here. And in our cartoon world here, the idea is the chromosomes have been found by the microtubules, and the microtubules have helped to line them up. It's as if the cell is taking roll, making sure all chromosomes are present and accounted for. Now let's take a look at the first two stages of mitosis using some stop action. Here we see some microtubules undergoing some random exploration of the cell. Sometimes they're growing, sometimes they're retracting. And now let's put some chromosomes in the mix here. And so now we've got some chromosomes are going to show up here, and the microtubules are going to explore the interior of the cell to find these chromosomes. And that's a trial and error process. The microtubules do not know where the chromosomes are, but when they make contact, they can help to line them up. Now in the third stage of mitosis, of course, those duplicated chromosomes are going to be separated to opposite sides of the cell. Here we see two nice cells caught in the act. And in our cartoon version here, the chromosomes, these would have been two duplicated chromosomes. So these are identical chromosomes that have now detached. One's going to one side, the other's going to the other, and so too with all the chromosomes in the cell. What that looks like for the stop action is something like this, where we have separation of the chromosomes. So in the final stage of mitosis, the division stage, we have the chromosomes are widely separated in the cell, and then the cell will divide down the center here. Here we see, if it's a plant cell, you'll have to grow a cell wall down the middle there. If it's an animal cell, it'll just be the membrane will pinch in here. On stop action, it looks something like this. Notice the nuclear membrane will reform around the chromosomes. Now let's take a look at the whole process with this version. We have microtubules exploring the interior of the cell in a trial and error fashion, looking for the duplicated chromosomes. And when they find them, they help to arrange them and line them up. And now the duplicated chromosomes will detach, and they will be separated to each side of the cell. And then a nuclear membrane will form around each set, and the cell will divide into two. So mitosis, in summary then, is a four-stage process where duplicated chromosomes are condensing and then being delivered to two daughter cells. And this is the nature of reproduction. Chromosomes are composed of DNA, and DNA is the hereditary molecule for life on Earth. Mitosis makes sure that one cell can make two cells, each with the full set of chromosomes necessary for life.